ladies and gentlemen, hosted by Korea Health Industry Development Institute. Welcome to BioKorea 2020 Invest Fair. I am announcer Hong Seon, today's MC. This event is to introduce pharmaceutical and biohealthcare companies' core technology, as well as to share their strategies and vision to potential investors. This session is for Rznomics. Rznomics is based on RNA platform and it works on anti-cancer therapy. It targets liver cancer, so it is currently preparing for the first clinical trial. Let's listen to the details by President Lee Sung-wook of Rznomics. Good to see you, President Lee. So a lot of people wonder about Rznomics. Please begin your presentation.
Sure, I'll begin my presentation. As you, Anchor Hong, said, I'm President Lee Sung-wook of Rznomics. Our company focuses on RNA platform to come up with an RNA new drug. So I'll begin with the introduction of my company and then move on to platforms, technologies, and our R&D pipeline, as well as intellectual property and business development plans. Rznomics, as you can see from the name itself, our title has our meanings. It's a combination of ribozyme and omics. So it's a transparency ribozyme. We have this platform technology. We focus on cancer and those intractable diseases, and we focus on this targeted DNA, and we target those RNAs to switch with this ribosome, and we make a DNA form virus platform to deliver it into the human body to cure liver cancer, intractable cancers, degenerative diseases, as well as genetic diseases, to meet those unmet medical needs, to come up with a gene therapy. Our company was founded late in August 2017, so it's basically a new company. And I am professor at Dankook University's Life Science and Convergence. So our headquarters is located in Dankook University, and we recently moved our research center to Pangyo Innovation Valley. Let's move on to our members. It's mainly, it can be subcategorized into three. Director Jinju Kim is in charge of management planning, and Director Sung Woo Hong is in charge of clinical development, and Director Sung Yeol Han is in charge of R&D, and we have legal, labor, and accounting sectors as outsourced. And also, we have Professor Jung Jin-suk and Professor Sang Yun Han and Professor Bruce A. Soliger as outsourced clinical medical advisors for our company. Let's move on to the platform. The transfer splicing ribozyme is a gluon introin based ribozyme, and we developed this therapy. In nature, this global neutral, as you can see on the left slide, this is a transsplicing ribosome. This means that without any external need, this entity can delete its own self and connect this through radiation. And if we could artificially connect these two RNAs thanks to radiation reaction and make it this through this ligation technology, we can come up with this engineering technology, and by using this transparency technology, we can designate this specific disease-related RNA, and we target this and get rid of it, and we use this therapeutic RNA and attach it to the end of the ribozyme to switch it to this part, so those specific RNAs can be expressed as wanted. So we come up with therapeutic approach. And in fact, the concept of this was first found in about 20 years ago, but when this idea was suggested about 20 years ago, as you can see here, only 6 ribozymes were combined, so the accuracy and efficacy was really low, so they couldn't clearly come up with a cure. But over the past 20 years, and when I was working at Dankook University, I learned that these target-specific characteristics was something to target on, so we have been developing on this, and in order to increase efficacy and effectively, we worked on this transferring R&D optimization. So that is why we founded Rznomics. So all the transversion ribozyme is when it comes to this skeletal cell anemia, among other genetic diseases, can be treated with this, and where viruses or cancer, among other diseases, related target RNAs could be targeted, and we use this antivirus genetics, or those cancer occurring cells can be replaced thanks to our technology. And this slide shows the global development, which is based on nucleic acid. It's DNA based and RNA based and gene editing technology. And those companies who have these technologies have their own characteristics, and they can optimize uh, by coming up with their drugs. And our main goal is within a few years, we use this trans-splicing ribozyme to come up with the therapy, which is our ultimate goal. And when it comes to this ribozyme, these features can come up with a lot of features, healing features. However, instead of that, we want to specifically focus on those unique characteristics to effectively target those non-targetable diseases to reach and optimize our technology to come up with the therapy. And the strengths of our technology are explained here, and there are specific targeting of disease-related RNA, which means we can target those diseases. And as I have mentioned, one molecule can inhibit a target RNA, and at the same time, we can specifically deal with those treating therapy molecules. So we can take a multilateral approach, and as I have mentioned in the former slide, those transversion RNA and a therapeutic RNA can be expressed, and the enzymes can be expressed. So they are all classified into different categories, which means we can take various approaches on respective targets, and they can come up with a module, so we can easily come up with an engineering technology to designate different targets, so we can make use of that characteristics. Let's move on to the pipelines of our company. The pipelines are shown here. We can majorly categorize into RZ001, 001 plus, 002, 003, and 004, mainly 5. So as for RZ001, we have a radiation combined pipeline. So RZ001 and 001 plus is mainly focused on liver cancer, and RZ001 is for glioblastoma, and 003 is Alzheimer's disease, 004 is retinitis pigmentosa, among other genetic diseases. So far, among a lot of pipelines here, we are mainly advancing here at RZ001. We have came up with a clinical trial therapy, and we confirmed the efficacy and safety. Currently, we're under production. So in this first half of 2021, we aim to go through for IND profiling, and we are working on this combined therapy with 001. Afterwards, 001, we will apply for an R&D profiling. So I will mainly focus on RZ001. As for RZ001, as I have just mentioned, this is targeting liver cancer. And a few years ago, sonafudib was the almost the only therapy, but nowadays a lot of ENFTAs are getting approvals, and in the near future, there are a lot of new drugs. And those combined therapies, especially for immunotherapy, will likely form the market. And our company's targeted DNA is this telomerase extending the end of the chromosome. Not only liver cancer, but almost 80 or 90% of other cancers has to do with this HTERT's expression. And not only liver cancer, but other cancers has to do with this, which is well known. And specifically for this liver cancer, the term of mutation, from the very early stage of cancer, this can be found. So throughout various pieces of research, we learned that HTERT is the main factor for liver cancer, which is well known. But when it comes for this telomere and targeting, there are a lot of weak points. For example, the most renowned weak point is especially for stem cell. The leg is really long, so we can't ultimately kill the cancer cells. So in order to kill a cancer, then you need long leg period. And another one is that, as I have mentioned before, liver cancer is often followed by liver cirrhosis, and those non-tumorous liver, there are livers, and in terms of those livers, they react to telomerase. So as for telomerase deterring agents can be targeted by this entity. So in order to target, not only should we deter TERT, but we should take multilateral approaches. And as for normal livers, we have to take care of the dysfunctioning of it. So we have to minimize the side effects. So our agent can overcome those weak points. So we have high expectations. As you can see on this slide, especially for cancer cell, we can recognize this TERT and we get rid of TERT RNA like this. And instead of that part, we can use this specific TERT RNA by using these therapeutic genes by transcriptional ribosine therapy. And this therapeutic RNA that we are targeting to develop, we can use HSVK, which stands for herpes simplex virus thymidine kinase. And as for this, there is a gencyclovir drug, and by transforming that into this and by using this bystander effect, we can ultimately kill cancer cells thanks to this. And we make use of this ribozyme and make DNA shape to deliver this. And we use this delivering vector, and thanks to our research, we learned that adenovirus is the most proper vector as a delivery channel. And as for heterogeneous cancer cells, we can also target them, and I'll elaborate this a little bit later. But thanks to this, we could come up with this improvement. And another one is TERT, and not only for cancer cell, it can be found from other cancers as well. So we can expand our adaptability to other fields as well. And this is what I have mentioned in the very early stage of my presentation, but for transversation ribosome, when that is limited, the efficacy and specificity is really low. So throughout the research from the past, as you can see here, the target RNA can be combined from these 6 of these, and also we can use this internal guidance sequence, and the antisense sequence is adapted here, and you can use this input P10 to enable this and optimize this therapy, and highly efficient transportation adaptation can be optimized and realized. And especially for this liver cancer, you among those symptoms, we can use this to show anti-cancer therapy. So based on all the research that we conducted, and in fact about 6 years ago, there was a researcher-led clinical trial on progressive liver cancer, and back then Professor Han Samyan's team in Donghua University got this IND approval. And based on the past research, we learned that in order to adapt this to liver cancer patients, we need to overcome a few weak points. That is because of this immunogenicity and the efficiency, efficacy, and capacity has to be optimized. And another thing is that this agent itself, as for normal liver cell, those normal liver cells could be targeted as well. So um, as for normal liver cells, we should not target them in order to increase safety. So far throughout the research that we have conducted, so to mention it again, as for this SDSA and WPRE, we had to additionally administer this agent to, even if we use only 10% of this virus, we could come up with 100% efficacy. And in order to prove safety, we had to study this microns 120 target sites optimization. So by this, we realized this for cancer cells, but not on normal liver cells. So we should target this only on specific cancer cells, so we can sustain our efficacy. So we have realized that optimization. And based on this agent, we selected candidate agents, which is called RZ001. And as for RZ001, as you can see here, we proved the efficacy case. So even with small dose on the day, and we can come up with intrahaptical and orthotypical. We can kill the cancer cell effectively. And another one is that systematically, when we delivered this, there's a VD data, we could acquire this. So as you can see here, within 2 weeks, in the blood, the virus was killed, and even in the liver, the virus was killed by 70%. And another important point here is that in order to increase the efficacy of the virus, besides increasing the efficacy, another thing is that by using the best delivery route, we decrease the dose of virus. And so we came up with this Hepart Rightry, which is the major supply of liver cancer. So we used the cirrhosis associated HCC to deal with hepatic artery and a vein, and we studied the distribution model. And as for in vein, the distribution level was almost identical, but as for the left graph here, as for liver cancer, it showed almost fivefold of accumulated data, so we could define it. So when you take a look at this side, just like the vein, when you target liver, liver is mainly targeted, but within 2 weeks, almost 95% was distinguished for not only VD, but chloride results was also found. And as for TK expression, what is important is, as you can see from the slide here, as for liver cells, the immune cell infiltration was really high, and secondary and challenging experiments were conducted. And as you can see here, those challenging cancer cells were almost 100%. Those adenovirus transversion was deterred. So which means that in terms of this gene therapy and transversion albozyme and targeting albozymes helped us to increase this local systematic immunity, so it can increase your immunity. So in terms of the efficacy of increasing optimicity, this can be combinedly used with an inhibitor cell. So this result implied that. Can we use RZ001 on liver cancer patients? So we wanted to figure out. So we conducted this research and we screened this. And besides those HCV patients, most of those non-HCV patients showed that the cancer level decreased massively. So this RZ001 approach was for those HCC related cancer cell patients, but most cancer cells. We learned that RZ001 could be used. Likewise, I have just finished mentioning RZ001. Let's move on to RZ003 and 004. RZ003 is based on RAR001 and is targeting Alzheimer's disease. As you know, in fact, recently the A beta, which is amyloid beta, targeting immunotherapy approaches recently turned out to be failures mostly, and nowadays new targets and new agents based research is working on, and there are various viewpoints saying that we need a early stage targeting therapy. And here is APO, which is apoprotein E, which is a protein agent. We target this APOA to adapt our technology through this approach. This APOA specifically has these three genotypes, and specifically APOA4 in terms of amyloid beta and its accumulation or tau's hyperfolation or APOA's toxicity protein accumulation or accelerating BBB breakdown. In considering these characteristics, APO4 acts as a risk factor of Alzheimer's disease. So especially for those patients who have this factor, have a much higher percentage to get Alzheimer's disease, and the time of getting this in terms of time and age, they showed a higher rate of getting Alzheimer's disease. So we targeted this risk factor APOE4 to decrease the risk factor, and we specifically targeted this benefit factor to realize that and express it to deal with Alzheimer's disease. And we delivered AAVA and delivered it through cerebral spinal canal to come up with this therapy, and we currently is working on optimizing this. And last is RZ004. So RZ004 is when you lose your eyesight, when you go blind, you may suffer this retinitis pigmentosa, so we target this with our molecule. So as for this retinitis pigmentosa, a lot of companies are currently working on this, and especially to deal with recessive types. They take a white type approach to deal with this and come up with a therapy, but in fact, our company's agent compared to other agents has a competitive edge, especially for retinitis pigmentosa, because this retinitis pigmentosa, from now on I'll call it RP, so from 15 to 20% is autosomal dominant mutation, which means that autosomal dominant phenotypes means that not only simply putting or injecting Y type, we have to get rid of those agents as well. So for autosomal mutants, targeting approach is almost currently, there are only one or two clinical trials working on. So our approach, as you can see on the slide here, on the left, the autosomal mutant related messenger targets, we get rid of these mutants and we inject ytRNA, and the RNA level we modify the DNA, and through this not only do we express this Y type gene, but also we deal with mutant RNA. We get rid of this, so basically we catch two birds with one stone, and we we attach it to AAV virus and we target inject this to deal with the disease. And let's move. I have just mentioned the pipelines, and now let's move on to the pipelines. So most of our IP is focused on RZ001. We have patents and applications, and we have 003 and 004 under application. So recently, September last year, we got Series A investment, and we had preclinical and clinical research working on. But later this year or early next year, we will have a next round of funding to come up with combined therapy to work on clinical trial in Korea and overseas, and work on pipelines and manufacturing to strengthen our capacity. The last slide shows the partners, the companies and research centers, and non-clinical and clinical research centers. That is it. Thank you for listening.
We have listened about the details of our genomics. So we'll look forward to seeing further advancement. Thank you, President Lee, for your presentation.